Hey guys, hope you're doing well. Today we're going to be discussing dominant and codominant genes in ball pythons. So get ready, let's dive in. I want to show you a dominant gene. This is a spider. One characteristic of a spider is clearly seen in its reduced pattern compared to a normal. This particular snake has other genes in her that contribute to her colors, but for the purpose of this portion of the video, we'll just focus on the fact that she has the spider gene and ignore the other color genes for now. Let's see how a spider can pass down the gene to its offspring. We can do that with the Punnett Square. Now that we have established that animal as a visual spider, we can represent that with the uppercase S. Since genes must come in pairs, we'll need an N. So now we have SN. If you watch my last two videos, you might be thinking that in those videos, when an animal has an N and an A, then it's a normal. That was true because the A was a recessive gene for albino. Therefore, the N overpowered the A. But in this case, we're talking about dominant genes, and the dominant gene S overpowers the N. So even having one S allows a snake to show visual signs that it is a spider. Alright, so let me write that again. So now we have our visual spider, and let's pretend we want to pair that with a normal ball python. Again, we represent normal as two N's, and we'll plug this into the Punnett Square. The male goes on top, female goes on the side. And we'll just factor these in. So we have S, N, 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 S, N, N, N. Each quadrant represents 25% of the offspring population. Adding up the percentages of like genes, we get 50% will be SN and 50% will be NN. As you can see, the offsprings that have S will be spiders, and the ones that do not have an S will be normal. Don't overthink it. The takeaway here is that when dealing with dominant genes, you only need one gene out of the pair to change the appearance of the snake. Even if the snake has two copies of the same gene, it will still visually appear the same as an animal with just one copy of the gene. As a side note, there are genes that are considered lethal genes. When working with lethal genes, you want to avoid breeding animals that may result in offsprings with two copies of the lethal gene, because it can cause physical and/or neurological disorders of the animal. The spider gene is somewhat considered lethal, so don't breed two spiders together. If you end up with an animal of SS, that is considered the lethal gene, and there's no benefit to producing an animal with two S's because it looks exactly the same as an animal with just one S. Let me show you some codominant genes. This one is a GHI pastel. The GHI is a codominant gene that really darkens a snake colors and busy ups the pattern. She also has the pastel gene, which reduces some of the brown and brightens things up. Over here we have a Mojave orange bee. The Mojave gene helps with the contrast and the color, and the orange dream colors up the snake. Both of these genes, when alone, make very subtle changes to the snake, but combined together, they work very well. So codominant genes, like dominant genes, only require one copy to change the appearance of the snake. But when the snake has two copies of the same codominant gene, it changes the appearance even more. When an animal has two copies of the same codominant gene, it is referred to as a super. All codominant genes has a super form. Some codominant genes are also lethal, such as the cinnamon and the black pastel. They are codominant genes, but when you put two together, it's possible to cause physical deformities, so you should avoid them. There are a few codominant genes that can result in leucistic animals in a super form. For example, the yellow belly codominant gene, when it is in its super form, is a completely white snake with black eyes, and it has adopted a designer name called Ivory. Let's get back to using the Punnett Square and look at a breeding scenario with two animals of codominant genes. You've seen the GHI pastel already. Both GHI gene and pastel gene are codominant genes. Over here is a firefly, which consists of two codominant genes, the pastel and the fire. Let's use Punnett Square to see what we can expect of the babies. So let's consider what we would get if we paired that GHI pastel to the firefly. So over here we have the firefly genes. This is the fire and the pastel. And then over here we have the GHI pastel gene. That's the GHI and that's the pastel. Male and female. So now we have the 16-cell Punnett Square grid, and we're going to plug in the parents' genes on top for the male and on the left for the female. So how we do that, again, we would start here. We have the F with a P, and then the F with the N. Then we have the N with a P, and we have the N with the N. Over here for the female, we do the same thing. We have a G with a P, then we have the G and the N. Then we use the other N here with the P, and lastly the N with the N. Alright, so let's go ahead and fill out this Punnett Square, starting up here with GP on the left and FP on the top. We just combine that, G, P, P, F. I just like to put the common letters together. So now we move over to this cell. Again, we're using GP on the left, and this time FN from the top. So G, P, F, N. And then we continue that over to here, G, P, P, N, G, P, N, N. Now we're moving down to the second row. We're using GN on the left, FP from the top, G, N, F, P. Moving over here, G, F, N, N. Again, I'm just combining letters that are the same, but they are still all here. G, G, N, N, F, F, N, N. Moving on to the next cell, we have G, P, N, N, G, N, N, N. We have N, P, P, F, N, N, P, F, N, N, P, 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 N, N, N. Last row, N, N, F, P, F, N, 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 P, N, N, N. Lastly, this one is completely normal. So here are the results, and because both parents have pastel, we would be expecting some offsprings to be super pastel. So the cells with PP are the ones that have the super form of pastel, and those are going to be seen here underlined. So we got 1, 2, 3, 4. There's going to be 4 out of 16 offsprings that have super pastel. Those are the odds. 4 out of 16, or otherwise known as 1/4, will have 2 pastel genes depicting that it is a super form of pastel. Then this one is going to have all 4 of those genes. So we got GHI, the 2 pastels being a super form, and then the fire. So this animal is going to be probably what we're shooting for in this pairing. And as you can see, the odds of hitting all 4 of those genes is 1 in 16. So that's going to be pretty tough to hit that, considering the female may not lay 16 eggs. So that's probably why some of these animals with multiple genes are very sought after. So this one again has GHI, pastel, pastel, and fire. Okay, so that was a pairing with animals that have all codominant genes. Now let's see what happens when we breed animals that have codominant and dominant genes. Earlier you saw my Mojave Orange Dream, and at the beginning of this video you saw my Orange Dream Bumblebee. So let's see what happens if we breed those 2 animals together. But before I start, I have to tell you that the Orange Dream Bumblebee actually has 3 genes. It has Orange Dream, Pastel, and Spider. I haven't showed you beyond the 16-cell Punnett Square yet, so just to keep things simple, we're going to omit the Pastel from the Orange Dream Bumblebee. So let's pretend she only has 2 genes, the Orange Dream and the Spider. So first off is the Mojave Orange Dream. We're going to show his genetics by MN, which stands for Mojave and normal, and then Orange Dream, so it's Orange Dream and normal. Now we're going to show the Orange Dream B, so that would be Orange Dream, normal, and it has the Spider gene, and then we're also going to consider it to have a normal gene there as well. So now, again, we have to have our 16-cell Punnett Square. We're going to put the male's genetics across the top. So I have M and O, and then we have M and N. Then use N, O, N, N. Now the female goes along the side on the left, so we have O and S. Then we have O and N, N and S, N and N. Alright, let's go ahead and fill in all the cells. So starting at the top right, we have S, O, O, M, O, S, M, N, S, O, O, N, soon, O, S, N, N. Next row we have O, O, oops. So again, I'm just combining letters that are the same to make it easier for myself. We have O, O, and N. That's a super orange dream there. O. Orange dream normal. Next we have S, M, and then there's S, O, and then normals. We have S, normals, Mojave, Orange Dream, Mojave, single gene, Orange Dream, single gene, and lastly, the normal. Here in the results, the OO represents the super form of Orange Dream, and those are really pretty orange colors. But the real prize would be the Super Orange Dream Spider, which I have boxed here in red. The odds of getting this one is again 1 in 16 getting a spider super orange Mojave. So remember we had omitted the pastel gene from the orange dream bumblebee? If we didn't remove the pastel from the breeding equation, then there's a chance we could produce a Mojave pastel super orange dream spider. So let's take a look at finding the odds of hitting that snake. Well, we know she only has one pastel gene, so we have to represent her with a P for the pastel and an N for the normal because they come in pairs. We know since that she only has one P, she can only pass down that gene to half of her babies. That means 50% of her babies, or it can also be represented as 1/2. The other half will get, the other 50% will get the normal gene, so we're not concerned there. So I've crossed that out. Now, in order to find out the chances of getting the Mojave Pastel Super Orange Dream Spider, we simply have to factor this P, this side of the equation, back into the Punnett Square, the 16-cell Punnett Square. But since we're only looking for that one particular animal, we don't actually need to work through another set of Punnett Squares. All we need to do is multiply the half by the 1/16 up here. So if we remember to do our math, in order to multiply the fractions, we just multiply the denominators and we get 1/32. 1/32. Out of a clutch of 32 eggs, only one will be a Mojave Pastel Super Orange Dream Spider. Those aren't very good odds, and since ball pythons do not produce 32 eggs a year, it's rare to get babies with so many genes. So if you're breeding for that 4 or 5 or 6 gene animal and you don't hit it right away, I hope you now understand why, and you shouldn't feel frustrated. Knowing more about genes will provide you with some realistic expectations going into each breeding project. So now let's go back to looking at the odds of hitting that special animal. The odds is 1 in 32 of getting a Mojave Super Orange Dream Pastel Spider. Let's prove we're right. So in order to do that, we have to use an even larger Punnett Square. So before we get into setting up that larger Punnett Square, let's just take a look at the actual genetics of the Orange Dream Bumblebee. So like before, we have Orange Dream. Genes have to come in pairs, so Orange Dream and normal. And then we have the Spider, normal. And lastly, we have the Pastel and normal. So genes always come in pairs. So this is now going to be the female that we're going to breed. And again, we're going to use the Mojave Orange Dream. And we're going to breed that to this female. So the Punnett Square we're going to set up is going to be much larger this time. It's going to be 4x8. So instead of putting the male up top and the female on the left, because the way I drew the grid, I'm going to switch it up. I'm going to put the female's genetics along the top and the male along the left. So the male's genetics is pretty easy because it's going to be the same as before. So we have MO, MN, NO, and NN. The genetics for the female along the top. So this is going to be new to you. We're going to start with the first letter, of each pair, so that's O, S, and P. Now we're going to do the first letter of the first two pairs, so O, S, and then the last letter for the last pair. Now we're going to use the first, last, and first, so that's O, N, and P. First, last, last, and now it's last, first, first, last, first, last, 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 first, last, last, last. Now we get to fill it in. Again, we're just going to combine what's on the left with what's on the top to fill in that specific cell. So here we have M, O, and O, super orange dream there, spider, pastel. So there it is. That's the one we're shooting for. And I'll just go ahead and fill in the first row. So we have Mojave, orange, super orange dream, spider, Mojave, super orange dream, pastel, Mojave, super orange dream, Mojave, orange dream, pastel, spider, Mojave, orange dream, spider, Mojave, orange dream, pastel, Mojave, orange dream. And let me go ahead and fill in the rest. So here we are, all filled out. As you can see, the first cell is what we're shooting for, this Mojave super orange dream pastel spider. And since this grid has 32 cells, 8 across, 4 down, that's 32, which means 1 in 32. The odds of hitting this is 1 in 32, so we've just validated that what we did earlier. And just for fun, we can take a look at the odds of hitting a super orange dream. So all the ones that have the double O's, 1, 2, 3, 4, 5, 6, 7, 8. So the chance of hitting a super orange dream is 8 out of 32, which equates to 1 out of 4. So there's a quarter of a chance of hitting a super orange dream. And if we look at the smaller Punnett Square, the 16 cell Punnett Square, the odds of hitting an orange dream here, 1, 2, 3, 4, is also 4 out of 16. 4 out of 16 equates to 1 out of 4. So regardless of how many genes you're dealing with or how big of a Punnett Square you have to create, the odds will always be the same for that specific gene you're trying to shoot. So orange dream, 1 out of 4. Chances here of hitting orange dream, also 1 out of 4. The chance of hitting the Mojave super orange dream pastel spider is 1 out of 32. And earlier, our little cheat way of doing it, we also get 1 out of 32. If you find this video helpful, give it a like, and in the next video, we'll take a look at allelic animals such as the blue-eyed Lucy and one of my favorites, the Candino. If you want to be notified, hit the subscribe button, and I'll see you next time.